Yes, as the voice remembered, the meeting is recorded, so if you don't like to be on the video, please switch off your camera. Also, please remember to keep your mic off during the presentation. It is my pleasure to introduce today's speaker, which is Leonardo Caproni. Leonardo earned his PhD from the University of Perugia in Italy, and during his study spent some time at the International Center for Tropical Agriculture in Cali, in Colombia, working mainly on agrobiodiversity and nutritional traits in legumes. After that, he worked on several European projects, again aiming at promoting agrobiodiversity. And since 2022, he is an assistant professor at the Institute of Plant Sciences of Sant'Anna School of Advanced Studies in Pisa. He works mainly in understanding the genetic control of traits related to photosynthetic efficiency in both maize and barley. But he also has a broad interest on agrobiodiversity, especially working in African countries such as Ethiopia and Mozambique. And I think from the title of the talk of today, he will speak about some of this work. The talk title is Tapping Ethiopian Barley Diversity for Climate Change Adaptation and Resilience in Smallholder Farming Systems. And without further ado, I leave the stage to Leonardo.
Thank you very much, Chiara. I share my screen. Hope everybody can see my presentation. If so, I think I can start. Okay, so I'm really glad and happy.
Leonardo, it's not loaded up. I can't see your presentation.
You can? Okay, so I think I have to switch. Okay. I think now it should be fine.
Perfect.
Okay, thank you. So I'm really happy myself and we as a group to have this opportunity to tell you what we have been doing these last 4 to 5 years about our research on barley. Okay, before I start, I also want to specify we are not proper like barley people. We are most of us are plant geneticists. We work with quantitative genetics and also a bit of plant breeding, I would say, with many cereal crops, barley, maize, teff, Agrostis teff, and other cereals. Right before I jump into the presentation, I think was good to spend a few words about the Institute of Plant Sciences and Scuola Superiore Sant'Anna, which is the university from which I come from and from which I'm talking today. So we at the moment I am in Pisa. Pisa is of course known for the very famous Leaning Tower. But from a scientific standpoint, it's also quite important because it has this tradition. It's the birthplace of Leonardo, Fibonacci, and Galileo, and still today is an important university and research hub. We have 3 universities, the big one, the University of Pisa, and the 2 honors colleges, which are Scuola Normale Superiore and Scuola Superiore Sant'Anna. So these are, they work as independent university. We have our own rectors, both Scuola Normale and Scuola Sant'Anna, and actually we are like very small compared to the University of Pisa. Pisa is also there's the headquarters of the Italian National Research Council, as well as the Italian National Institute of Nuclear Physics. So if you put together staff and students, almost university and all this research hub matches the size of the town, of the city. So when you go out for work, you always stand up, you have high chance to end up talking about work and sciences and research. The Scuola Superiore Sant'Anna, so is this small university, which is research-oriented, public, is accessed by merit, just 50 freshmen per year on applied sciences, plant biotechnologies, biorobotics, some specific disciplines of engineering, medicine, law. and so on and so forth. For plant biotechnologies, actually, there are only available 4 positions for undergrads per year, so very few. And so the selection process is quite tough. Actually, there are more opportunities in terms of PhD's positions. This is our research group at the Institute of Plant Sciences. There are, I would say, 4 big groups, and this is the group of translational plant genomics. The first row, you see the faculties, and on the top left, Matteo, our current PI and full professor in plant genetics, and then all in second and third row, the PhD students. As you can see, we are coming from all different parts of the world, but not only this, it's also important I want to show this picture because many of us, we have different backgrounds. For instance, you see people highlighted in red are experts in plant genetics, genomics, or molecular biology, while people highlighted in green in socioeconomy or agriculture, agronomy, and people highlighted in light blue are climate scientists. And this is quite important to introduce what I will talk about today, our research, which was done on barley and in Ethiopia to study adaptation. So first question to answer, why we choose barley to study adaptation and adaptation to climate change. Well, I'm sure the barley community is very familiar with this paper. Now it's 6 years old, but still a great work, which is actually telling us that if we look at an horizon of 80 or 100 years from now at several climate change scenarios, we should expect to observe a global decrease in yields for barley. So it's very important to act in these regards. So we believe that looking into tapping into the genetic diversity, it can be still a valuable approach to look into possible allelic variation for ways that can help us adapting our barley in the future to climate change. And for this reason, we started looking at Ethiopia. So why Ethiopia? For us, geneticists and biologists, this is a quite special place because it's a place in which we can find many endemisms. There is this huge topographic variation in a relatively small latitudinal and longitudinal range. We have this topographic variation. It translates into lots of environmental variation. We can find 10 ecosystems, 31 registered agroecologies in the country. And when we look at crops, it's also very interesting because in the country, agriculture represents 50% of the GDP, and most of agriculture, it's still today, 2024, conducted by smallholder farmers. I have this figure is telling us 90%. It may be slightly different now. This probably data that goes back like 10 years ago. But it's roughly representing what is agriculture now in Ethiopia, and so 80% of the total workforce of the country of Ethiopia works directly in agriculture. So in Ethiopia, barley is a staple crop. It's very important culturally, and this history goes back almost 5,000 years ago. There are archaeological findings that testify its use and cultivation 5,000 years ago. And it is found in a broad range of agroecologies, especially between 2,000 and 3,000 meters above the sea level, where most of the human settlements are, where most of the agricultural activities are carried out. It is cultivated mainly as a kind of spring-type barley during the rainy season, which is called mehab, which goes from June to December. In many areas of the country, until 20 to 30 years ago, if you talk to farmers, actually it was also possible to cultivate barley during the other season, which is the dry season, which is called Belg. But it seems that in many areas it's not possible and convenient anymore because of climate change. Another very interesting fact about Ethiopian barley is, of course, its diversity, and I'm sure with like working with barley, the community knows very well this publication, which represents the entire big resequencing exercise, this is a sequencing exercise, genotyping exercise of the entire IPK gene bank, and every dot represents a different landrace. And here we see that the material coming from Ethiopia seems to be very, very unique compared to the others. So here we think we can find potentially allelic variation to study adaptation. So we were lucky enough to start this project because we had the opportunity to work with the Ethiopian Biodiversity Institute, which is the largest gene bank of Africa. So when we decided that we wanted to go ahead and study adaptation, the first thing we did was to assemble a diversity panel by selecting a number of accessions, in this case 249 georeferenced landraces, which were representative of the cultivation range and all the agroecology in which part is actually cultivated in Ethiopia. And as you can see in the map, every black dot represents a sampling point where these landrace accessions were collected not long ago, between the end of the 90s and the early 2000s. And this material was actually purified at the beginning of our work before going ahead and characterizing this material. It is known that in Ethiopia, farmers sometimes tend to mix like different materials, and in fact, when you like see how these accessions are on a field, you start observing that within the same accession, sometimes you find different morphotypes. So our first big work was to separate these morphotypes and to split these accessions and start a process which was quite straightforward, using single seed descent and amplifying this material. And so with this purification process, we ended up with 383 genotypes, starting from 249 georeferenced landraces. And we actually decided this panel of genotypes with 40 varieties, improved varieties, which were released in Ethiopia at the time of the creation of this panel. And all these improved varieties, they were released and developed by Ethiopian institution, private or most of the time public institutions. Actually, the experiment consisted in two common garden experiments, which were repeated in 2 years in full reps with alpha lactis design. And in these big experiments, we started collecting data about phenology, which is something interesting to look at when studying, of course, adaptation. In these trials, we also measured other characteristics of this collection, including also spike morphology, plant height, and also estimation of potential grain yield, but not really something we have published. And this was the big work done by Sant'Anna, which was PhD student at Scuola Superiore Sant'Anna at the moment, and currently is a research scientist at the Ethiopian Biodiversity Institute. So as we are geneticists, we actually sequenced and genotyped all this material using double digest restriction-associated DNA sequencing. So it's basically a genotyping approach in which we use a pair of restriction enzymes, and we select like certain parts of the barley genome. We select, of course, these enzymes based on in silico kind of mutations to optimize and maximize the number of loci we can type. And we ended up with a panel of 23,000 SNPs with a minor allele frequency measured at 5%. And actually, we also derived a panel of 2,000 linkage disequilibrium pruned SNPs to study the genetic structure and diversity of this material. So the idea is that we immediately saw that the improved materials were different. They set apart from the land races. Some are actually seems to be the product of mass selection directly from land races, which these are maybe varieties which were released in the past, and others seem really to set apart. And also looking at the pedigree of this material, are also bringing in, of course, the allelic variation from the international gene pool. But if we look at this kind of neighbor joining tree, we see that the regional provenance, which correspond to the colors here in the map, that does not really explain the structure and the grouping we observe. Yes, it does partially. For example, we see the group of Tigrinian accessions from northern Ethiopia that tend to cluster all together, but not really. So it seems there is something more to look at. So looking at the genetic structure, we use like multiple approaches. We use discriminant analysis of principal components, for example, and also admixture. And we had somehow concordant, I would say, suggestions that this genetic diversity could be best summarized with 11 clusters, so quite a complex genetic structure. And if we look at the map of Ethiopia, the different dots again represent the different land races and where the points in which they were sampled, and the colors are actually the different genetic cluster that we estimated using the APC. And we start seeing that actually there are latitudinal and longitudinal gradients which are seems to split groups. So not really administrative region of origin does not really explain, but we see something that is going on if we look at longitude, latitude, as well as altitude on the map. And in fact, if we look at the distribution of this different genetic APC cluster into different agroecological zones, we see that some groups actually tend to be found exclusively in some kinds of agroecologies. And so this is the kind of suggestion that is telling us that perhaps this actually material and the distribution of genetic diversity, a small portion of this genetic diversity and its distribution on the landscape may be explained also by adaptation. And so what we did was to go to the sampling sites, and actually we tried to extract climatic features and the sampling points where these land races were actually collected 10 to 20 years ago. And for this climatic characterization, we were very lucky to have on board in our group climate scientist Jamal, that you see in the picture, Jamal Sidamet. And with him, we were able to like customize and create this climate data set in which we only look at the months in which barley is cultivated at quite high resolution, high spatial resolution, and we actually use products which are publicly available, but we customize these products because we also had the opportunity to access data of 150 weather stations in Ethiopia, and to downscape, for example, the historical data related to precipitation using real world historical data which were registered in Ethiopia. This is actually very important to like produce this kind of custom set of climate data, because some areas of the world, like Ethiopia, in which we have this wide topographic variation, actually sometimes in some spots, in some areas, also the historical data are not extremely precise and do not really capture the real thing. So this was actually very important to start our study. So what we did actually with this historical climate data is that we derived 19 bioclimatic variables, bioclimatic indicators, which are actually indicators that are describing within this period, which for us is Meher, the distribution of precipitation and the different patterns of temperature. So out of these 19 bioclimatic variables, let's say that roughly half of these are describing temperature like patterns, and half of it like precipitation. We kept only climatic data in agroecologies in which we have quite a high density of data, so 10 at least 10 genotypes collected in a certain agroecology. Then you will see here the map. This is the union of all the polygons that are making this area which we are studying, in which we have actually samples, and we have genetic information of these samples. So at each of this layer, we extract all the information, not in all 19, using all 19 layers, because sometimes in certain areas, of course, these bioclimatic variables are extremely collinear. So what we did, we just randomly resampled points in this area, and we actually test for collinearity, and we use this approach which is called variance inflation factor analysis, in which we keep the minimum set of bioclimatic variables which are explaining the maximum portion of variance in terms of climatic variation. So now we have at each sampling point, we have layers of climatic information, several layers of climatic information. So in every point we have also genetic information, because we have genotype for our collection. So the first question we wanted to answer was: is there any method which we can use to understand how much is the proportion of genetic variance which is actually explained by climatic variation alone? And actually, we use this approach, which is used a lot in molecular ecology, which is called redundancy analysis, or partial redundancy analysis. So the idea is that we use this redundancy analysis, which is basically an approach which combines principal component analysis and multilinear regressions, in which we can use different sets of explanatory variables and test response variables, which for us are our genetic variants, our SNP, and in this case we use linkage disequilibrium prone SNPs. And which are and regarding our explanatory variables, we use altogether the bioclimatic variables, so these variables which are non-collinear, which are describing in every point the climatic variation at every sampling point for our landraces, but also their geographical position as coordinates and also the genetic structure. And if we use a full model, we see that overall we can explain with climate, geographical position, geographical coordinates, and genetic structure as the first three principal components of the genetic structure of our panel, almost 22% of the total proportion of variance. Now, in a stepwise fashion, we can test the effect of one single set of explanatory variables by correcting and factoring in the others. And this way, we understand that 5% of the total genetic variance, we estimate that actually can be traced back to climatic variation. So our hypothesis is now that there is a small proportion of genomic variance in this diversity panel, which actually is can be explained and can be related, can be traced back to adaptation. Of course, these are cultivated materials, these are land races, these are not wild materials, but somehow most of these variants, of course, should be the product of selection of farmers throughout generation, but there should be a little tiny trace of adaptation, and we estimate this small portion of adaptation is 5% of the variation we can observe. So we use another approach to model this portion of genetic variance that varies with climate, and we use this machine learning tree-based approach, which is called gradient forest, which is a lot used in ecology and landscape genomics approaches. And this time we use as response variables our SNPs, and as predictors our bioclimatic variables. What does it mean? It means that we want to build a function which is based on decision trees, on many decision trees, on forests of decision trees, and basically we predict the genetic composition at each point of the map by starting only with geographical position and climatic features of that position. So the idea is that we can transform, we can forget about our sampling points, which were used to build a function, and now we can project this genetic diversity that we estimate with this machine learning approach on the map. So what does it mean, this map? It means that each dot, each pixel, each point of the map is actually represented by a different color. And this color is actually, when the colors are similar, we expect that we should find land races with similar genomic adaptation locally. And so they should be similar genetically from if we look at their adaptation kind of features. And so it means, in other words, it may sound a little bit tricky, but we convert in every point of the map these bioclimatic variables into genetic and genomic composition, right? So it means that we also use, of course, geographical position, which is affect, of course, this pattern of adaptation we know, and we can also measure this. But it means now that we have this function that transforms climatic and geographical features into genetic composition, right? So now we can detach from and stop using the historical climate data, and we can actually plug in this model, in this function, projected data, future climate data, and see how we predict this genomic and genetic diversity distribution on the landscape. And what we see, actually you see here, the transition of colors and the pattern on the map is, and also the colors are quite different. It means that in the future we should expect actually to observe a different distribution of genomic diversity to actually explain like the climate in the future. And actually, if we can measure the discrepancies, the offset between what we predict with historical climate data and what we predict with future climate data, and we can estimate what is called a genomic offset, which is calculated as the median distance between the genomic composition of the future versus the past. And we see that in some areas we see like that these differences are very, very high. It means that in these areas what is cultivated at the moment perhaps in the future will be less adapted than material that is found in other areas nowadays. And so this doesn't have nothing to do, of course, with yield or potential performances of this material, but it just means that the distribution of genetic diversity that seems to be associated with climate in this part will not be the best to potentially compensate for the future climatic conditions. And actually this way, what we can do, we can actually estimate the optimal migration. So where should we like pick alleles or landraces to bring from a position to another in the map to compensate such genomic offset. And after we actually identify using this machine learning approach potential climate adaptive loci, we actually started looking into that also by comparing this loci with loci that we mapped using phenological traits. For example, here you see that we measure our diversity panel for phenology, I told you, and we tested GWAS, and of course we see that it seems that internalization H1 seems to play a very big role in the controlling phenology in the Ethiopian germplasm, as well as other loci which are associated with phenology also in other species. But if we actually look at the redundancy analysis and we identify the outlier loci, which is not a multiple regression approach as GWAS, it's actually different, even if the graph look very similar, we identify some loci that actually play also a role in phenology that actually seem to explain adaptation, and including other like loci that are like have been associated in barley, but also orthodox in other species to control phenology. So we focused our attention on Frigida, on this copy of Frigida, which actually has been reported also in other species to play a very important role in enhancing multivariate adaptation, especially with having a pleiotropic effect on many like phenotypes that can be like associated with adaptation, like escape strategies for dehydration, late flowering, and slow growth in Arabidopsis. And so the idea is that within this panel, we potentially have like alleles that can contribute adaptation. So as we are also very much interested in quantitative genetics, the first thought was why don't we select and capture this genetic diversity, and we actually combine and this genetic diversity to make a multi-parent segregant population, which can be used as a platform to map, of course, quantitative traits, but also can be a platform to develop pre-breeding material and test pre-breeding materials for future Ethiopian breeding efforts. And so we did select many improved varieties which have, of course, the Ethiopian like landraces in their pedigree, and also some landraces to make multi-parent cross design, multi-parent population. And this is actually the idea. This is, I think, this is quite new. I've never presented this population actually in a formal occasion like this. So this, we call it. I'm sure you are familiar with multi-parental population, and this is a kind of a design which bridges and brings together the advantages of a nested association mapping with a multi-parent advanced generation intercross design. So the idea is that we have 5 four-way magic population, which are all interconnected. This is iMagic. This is why it's called iMagic by a recurrent funder, which you can see here in the PCA. So again, this is the whole panel of Ethiopian diversity, and this you see all the different funders of the population, the recurrent, to make the 5 different families. And this was the big work which was done by Assetti Kassau, which actually made the population in this process was very long. It actually started before I joined the group. So actually, last year, so relatively recently, we flew to Ethiopia, and we actually went in Barda, nearby Lake Tana, and we extracted DNA from the recombinant inbred lines, which were developed with this iMagic population. Not sure it's okay. And so this was also a good occasion in Ethiopia in this lab. I mean, it was the first time for the lab to experience this kind of experimental activity. It was the first time that DNA was extracted in this laboratory, and so we were so lucky also to be in the first page of their public yearly publication. This was done in collaboration with Harare, which is the Regional Agricultural Research Institute of the region Amhara, in which Seyum is working, and also where Zeino, our PhD student, Zeino Tehat-Lapi, is actually, he was working before joining us for PhD. This is the preliminary data we have from our population. This is, of course, a PCA, which is representing the genetic constitution of this segregant population with different colors. You see the different recombinant timbres belonging to different subpopulation of this iMagic. And from a genetic standpoint, it's very interesting material because we see that has a relatively fast linkage disequilibrium decay when we look at decay versus distance. So it means that there is a high level of recombination. This is, of course, by design of the population. And we are still working on the making of the genetic map of this population to start QTL mapping. But actually, this population was phenotyped within the PhD project of Zino, the Hartsuafi, and in the framework of the project Capitalize, which we are working on several crops, barley, but also maize, for improving photosynthetic efficiency. And we had funds to characterize genetically, but also phenotypically this population. Here you can see a PCA in which we measure really basic phenotypes in two experimental location, full rep experiments for 2 years. So we have plant height, estimations of grain yield, 1,000 seed weights, based herring. And actually we see, for example, we are testing only using GWAS, not QTL mapping. These phenotypes here, it's a very simple binary phenotype, which is row type. Actually, we see that we have quite remarkable statistical power to map these phenotypes, but also more complex phenotypes like plant height. But these are just trials and preliminary data. But what was it's very interesting is actually that this population, it also seems to work very well to study very complex phenotypes. And actually the project Capitalize that I was mentioning before, where we are working together with Harari, actually we could measure some parameters related to photosynthetic efficiency using this portable device, which is called multi-spec, which actually is a pulse amplitude modulated fluorometer, which actually is able to measure re-emitted light, so chlorophyll fluorescence, and so we can have a fair estimation of maximum and operating efficiency of photosystem 2. Here you can see, for example, here the data that were collected at 2 experimental sites in Ethiopia with this portable device. And here you see like these measurements, which were taken randomly over the course of many days, the entire experiment randomly during entire days, in which you see like this data point measuring the operating efficiency of photosystem 2 in function of light and temperature, which actually is in collaboration with the University of Cambridge, thanks to Lucia Arce-Cubas and Johannes Kramji, we were able to actually model this measurements of the photosynthetic efficiency of photosystem 2, by fitting this third grade polynomial function, and by also measuring the genotypic effect as the residual variance, the deviation from these fits, let's say. And by using this approach, it seems that the population has the statistical power actually to map also these very complex traits. And actually, it seems also we map a region which was also reported in other experiments and other papers. Now I think I have to conclude because I think I'm going too far. But what we are doing.
Seem to have lost connection with Leo just now. Yeah, that's must be an issue at his end.
Yeah. Leonardo, can you hear us?
Hopefully he'll try to rejoin very quickly. There he is, back on now, I think.
So, can you hear me now?
Yes.
Yes.
Yeah, but we lost you for a few seconds.
I'm really.
I think we missed your last slides.
Okay. Not this. I want to share the other screen. Can you see the screen again?
Yes.
Yes. Sorry. This segregant population, we are trying to factor in farmers' appreciation. We are trying to use participatory approaches in order to collect quantitative kind of data regarding the appreciation of farmers. And the picture that you see, it's not actually barley. This is turunguis. This was done in some previous experiments, which are now published, and in which we actually used farmer scores to train genomic selection models to predict yield. And also we use these scores to map these kind of appreciations. How does it work? It's really easy. We collect, we do focus group discussions with this farmer. We ask them to evaluate certain traits which are discussed and agreed. And we ask them to evaluate in a Likert scale from 1 to 5 in these big experiments with replicates, so that we can also estimate best linear unbiased predictors of farmers' appreciation scores. And if we do so, actually, this is a very, very preliminary data we are showing, but we are able to map some of their appreciation. So you see here 2 different locations, Geragera and Abat, where these traits were collected, and male and females. These are data of 1 single year, but as you can see, in the same location, male and with appreciation score of male and females, we can map same loci, but also we can capture differences, which is really interesting. And also this is even more important when we compare data coming from different locations. So with this, I like to share with you some final thoughts, just to say that when we combine quantitative genetics and climate science, we can unlock the potential of land races, and by leveraging this diversity, we can put in place tailored strategies to also address the needs of smallholder farmer communities. This is to say that starting from this diversity, we could actually produce this breeding material, and we believe that together with institutional partners in Ethiopia, and by using a participatory approach, we can really provide good contribution to the breeding efforts in the country for a species such as barley. So with this, I want to on this project, in particular Bazazen Antum Kasso, which is the person behind the characterization of the diversity panel from the Ethiopian Biodiversity Institute, Zein Utehar Suafi and Seyu Masefi Kassau, which are actually 2 people which are behind the creation and the characterization of this segregant population. So thank you very much for your attention, and I'm open for questions. And I'm really sorry for the problem with connection.
Thank you. Okay, so it's open for question. I don't see any question in the chat at the moment. If anyone wants to ask a question, they can either raise their hand or unmute. Maybe I'll start with a question. Do you have, or are you planning to have any soil data? I don't know how easy that would be, considering it's Kenya, but especially because you're looking at photosynthetic efficiency.
Yes. So I think you are perhaps referring to the characterization of the diversity panel. So for the diversity panel, actually, so the photosynthesis characterization, we are carrying it out on the population, on the segregant population. And actually, we are also planning it would be optimal we are looking for resources also to characterize photosynthesis also on the diversity panel. And of course, when studying photosynthesis and when studying adaptation, it could be also interesting to look at adaptation in terms of factoring in different characteristics of soil. I'm not sure if this is what you mean, Chiara, but I guess that, of course, when you study adaptation and when you derive like environmental data, it's very easy also to include data of soil acidity, soil humidity. What we are really interested in, we were really interested in when we did this study, was really to look at adaptation to climatic feature. And of course, when you factor in many different and diverse environmental factors, and you start looking also at soil characteristics, then it gets really, really difficult to interpret what's going on. So it would be extremely interesting actually to look at these materials only by studying adaptation to different soil types. This actually would be absolutely interesting using these approaches.
I'm trying to look through if there is any raised hand. I don't see any.
There is one raised hand.
There is one. Okay, just
Hello.
unmute yourself and ask.
Chiara, can you hear me?
Yes, yes. Hi, John.
Okay. Hiya. I was going to ask you a question. A few years ago, we've actually had a PhD student working on an Ethiopian Eritrean barley collection that was developed by Brian Stephenson. And one of the things that we noticed when we went there about 5-6 years ago, we actually met Carlo when we were out in Ethiopia. And there was a big push towards growing barley for malting in Ethiopia. And some of the larger brewers, guys like Heineken, had sent over a couple of European varieties that they were trialing. And of course, they required a lot of inputs, fertilizer and herbicides. How do you, I mean, what do you see that as some kind of aspect of the work you're doing, particularly with landraces? Because obviously, you know, the landraces wouldn't be developed for malting quality, and whether this would be, you know, you're really looking towards just barley for food.
Yeah, this is an extremely interesting question. Thank you very much. So our assumption is that what we is present in in our diversity panel is only. So sorry, I hear some. Oh, okay, now so it's still cultivated by smallholder farmers. So what he's saying is that in the recent years there is a push in Ethiopia from big companies to produce malting barley, but this is not really affecting the smallholder farmers, which are the majority of farming activity in Ethiopia. And of course, this kind of farming activity, this kind of farming system is not really affected by this movement, this push from the industry. Of course, if you go nearby Addis Ababa, you see these big farms which are cultivating thousands of hectares with one single varieties, and these are producing malting barley for Heineken and kind of like brewing activities, but these are not really contributing to food security in general in Ethiopia. And barley, actually, it is in many parts of the country. It's a staple, still a staple food together with tap and durum. And I'm not sure how our work can help to find an intersection between what breeding companies are looking for in Ethiopia and the smallholder farming system. But what we believe is very important is that the design of a new variety in a system, in a complex system, agricultural system like Ethiopia, first of all, has to be somehow participatory. Somehow you have to co-design together with farmers the varieties can be more adapted to and to make the system a little bit more resilient in the future. It's not likely, from my experience, that if you go with a very high yielding modern varieties, these varieties will be accepted by smallholder farmers in Ethiopia, because many of them, and this is known and from study, are not really looking for yield per se, but these farmers are looking for many like traits which are not necessarily related to yield, and also most of these varieties that are introduced with these projects for to sustain this industrial development, they work really well with inputs, and most of the smallholder farmers, they don't have access to inputs in Ethiopia. So these are all evidence that are telling us that perhaps what we are doing is not really affected by this what is happening with barley in Ethiopia. I'm not sure if I answered the question. Perhaps it was a little bit too long.
No, that sounds good. I mean, I guess the thing is, you know, the Ethiopian government does want to lift a lot of people out of poverty, and one way, I guess, is, you know, if they can make more money selling barley for more, may well be that's, you know, something that we could combine with the, you know, like obviously the European lines are not suitable for growing in Ethiopia, but the combination of, you know, some of the traits from land races and potentially malting quality traits may open up a whole new area.
Absolutely, yes. But the big problem is that this process of introducing new varieties in the smallholder farming system, it's more complex than in industrial agriculture, which exists in Ethiopia, of course. Yeah, I mean, this is my perception. Actually, thank you.
I think I've seen Luke with the raised hand.
Hi, yeah, I really liked the talk. I was really struck by the way you were able to map this partial redundancy analysis back onto the genome to pull out regions you thought were involved in climatic adaptation. Could you go in a wee bit more detail there? And I was also wondering, did you pick up anything that you thought might be related to disease resistance rather than directly adaptation at all? I was just thinking for MLO, for example, or something like that.
Yeah, it's thank you for the question. So I think you are. I can scroll. I think I have some spells which I can use to tell you more about it. Yes, so the redundancy analysis actually it's let me see if I can find the slide. It's a combination of principal component analysis and multiple regression. So the idea is that we can transform all the genetic diversity, which is represented by all these dots in this RDA space, which is kind of principal component analysis. And then we can look at the distribution of the distances of these dots to the center of the RDA, and identify outliers based on a chi-square distribution. And so the idea is that this way we could actually map PPDH1 and Frigida back again, which is impressive, which actually using the same method and the same approach, the same loci were actually found in agrobiodiversity, which is quite impressive. And if we look closely to other like loci in linkage with these signals, yes, there are like gene models that seems to have orthologs that somehow are related to disease resistance, but this is very far to say that we are able to trace somehow back disease resistance. But if we look at the linkage blocks, which are in barley are always quite big, always. Where in some of for these signals we see enrichment in disease resistance kind of gene models, which is interesting. But this is something we actually did not discuss in the paper which is published, which we thought was a little bit too strong guess. it was too much. But yes, definitely it's absolutely interesting, and we believe that these approaches, if done properly with the right materials, actually can yield very, very interesting information. Yes.
Thank you. Cheers.
There's a question in the chat. Ruth, do you want to unmute and ask yourself? Or maybe I read it. So the question is, with the IBH population, how many phenotypes would you want to look at to assess yield stability, such as complex trait?
Okay, at the moment, within the framework of this big European project, which is actually at the very end, which is called Capitalize, we had resources to like conduct 2 years experiments at the location, and this was extremely tough because straight after COVID, there were the political situation in Ethiopia was really tough. Actually, in the northern part of the country, there was a civil war that then moved south. Also in the area in which we did DNA extraction, now it's not possible to go at the moment. And sometimes it was also very difficult to communicate. So there are all these things that are making everything very, very difficult. But we look forward to, of course, collaboration in the future to have resources to actually measure in multiple environments, multiple years, also very complex traits. We are really much interested in photosynthetic efficiency. The problem is that with these portable devices is that you actually to measure very, very big numbers, you have this huge environmental variability. So basically, in order to determine what is the effect of a genotype to the variance you measure, it's actually something extremely difficult. It means you have to measure and to produce thousands and thousands of data points. And this means that to measure very complex phenotypes and to be accurate in mapping these phenotypes, we would need a very, very high number of field trials and a very, very high number of portable devices and a high number of people working on it, which is still, at the moment, we don't have resources as a group. And something else which is interesting is also that the entire population and everything, there is no seed in Italy, everything is in Ethiopia. So this is really a strong collaboration with the regional or national institutions in Ethiopia.
I don't see any other question.
I think that's Luke.
Yes. Yeah, correct. Chiara, if that's okay, if I could ask another question.
Yeah, of course.
Again, of this iMagic population approach, which again looks very interesting. I was just wondering, you obviously had a mix of improved lines and land races. Do you have any thoughts on what's the optimal mix of diversity given the complexities of the genetics of the population at all?
Okay, this is a very interesting question. So sometimes when you do segregate population, you don't want to mix too much different genomes because then you would like observe deviation from expected, recombination. Now it becomes almost impossible then to trace stack recombination, to build genetic maps, and then to have this material, to use this material. Actually, what we see here is that we have selected a lot of the majority of the genotypes that are used to build this population are improved materials. You see the color triangles here. So most of it are, most of these are improved materials. And these improved materials actually, the selection of these founders was based on the pedigree of this material. It means that here you somehow also have alleles coming from the international gene pool, but also alleles coming from the Ethiopian pool. It means that here you already have genomes that are, you know, diversity is recombined. Also, you know, in the founders per se. And what we observe is actually that I only have, unfortunately, this PCA, but here, for example, you see this vector, not see of grain yield here. And we see that there are a fairly like important group of recombinant inbred lines that actually outperform the founder lines, which are landraces, but also parents. And when you have such indications, it means that the fitness is good of this material, and it means that probably the diversity we mixed was, you know, proper and not too much. Okay, so this is already a good sign. So if I would show you a distribution of grain yield, you would see that the founder of the population will distribute over this distribution normally. Okay.
That's impressive. No, thank you.
Thank you very much. Of course.
You're on mute, Chiara.
Oh, yeah. No, just to close this answer, it's also, of course, some of the lines are outperforming the founders. It is only based on 4 experimental trials. Of course, these are plots. These are full rep. The plots are relatively big, but of course, we need more data. But it seems like there are good signs that's like in terms of fitness, we have this even distribution of recombinant inbreds and founders.
And would these be used be released as varieties, you think, or?
No, I didn't get the question.
Could these be released as varieties, you believe, or do they need further breeding?
I think that this can be a good source of pre-breeding material. And I'm not sure if here there are lines that can be be used and to be released straight from the population. I know that Arari, in particular, Azeyu and Seyum that you see here in this slide, they are looking at a group of 20 recombinant inbreds that seems promising. And perhaps some of them in the next 3 to 4 years will be released. Yes, but this is, you know, they have to compare this material with what is actually. Yeah.
That's a real result. Congratulations. Cheers.
Yes, thank you.
Yeah, so I was saying, I don't think there are additional other questions, unless, yeah. I was saying that I was curious to see a follow-up of your male and female scoring on traits. And I remember reading years ago that the naked phenotype has been selected by women at some point because they were in charge of cleaning barley for human consumption. So I'd be curious to see that. Okay, so if there are no further questions, I think we can all thank again Leonardo for the great talk.
Thank you very much.
And I think Malcolm said before that the next seminar will be sometimes in January, probably middle of January after Christmas holiday. So I also take the chance to say, have a good Christmas holiday, everyone, even if it's very early, it's not even December. and I'll see everyone in January. Thank you.
Thank you very much. Bye-bye.